this medication with these old friends that we have now in the NHS and we can use to treat these patients, but also something that is coming up potentially, in the not distant future. I'm a pediatric neurologist. I work at Great Ormond Street Hospital, and I'm involved also in doing some of these clinical trials, so I have also this hat, looking at also what's happening in research. And this is the overview. So I will briefly mention, the 3 medications that we already have. I will give some minor, not comprehensive, possibly, overview of what's coming up, especially those that are probably more close to, to become potentially available in the near future, and also highlights again how important it is to treat early in this condition. So we all know what is spinal muscular atrophy. We know that the motor neurons in the spinal cord are the cells that are more keen to degenerate due to the low levels of the SMN protein. And this is a recessive condition where parents are both carrier in the vast majority of cases. And I like to show these slides because we have mentioned, Jeanette, this morning, the experience that it was fantastic. Thanks you so much for sharing. But actually, you can see probably, where we were. So the Wernicke-Hoffmann, disease was just described at the end of the 800 here in a few babies with SMA1, that we know now they are called SMA1, who died, for respiratory failure and so on, and they had this very peculiar aspect of the motor neurons in the spinal cord that they were generating. Then we had to wait, it was 78 that your daughter passed away. So in 95, we managed to identify the gene that was possible. At the time we still had to do the muscle biopsy. Now it's just a very quick genetic testing in 95. And in just 30 years, now we have 3 amazing medications that are working very well that we can deliver in the NHS. So amazing, more than 130 years, how the history of SMA has dramatically changed. Just a little bit of recap about genetics. So we know that children and adults with SMA lack both copies, you see here, of the SMN1 gene. But the lack of this condition, and this was the strength that was so critical in expediting, the development of therapies, was that in the evolution, in this region of the chromosome 5, there was a duplication that occurred naturally. We don't know why, probably because we needed more SMN protein. So there was a duplication of this region that led to the formation of this second, I call it the poor cousin, because it's not able to produce enough protein, so the SMN2, because only produces a small amount, 10–50% of the protein. But this is enough because all patients, all individuals have SMN2 copies with variable numbers, and we need at least a little bit of protein to survive because the lack of both genes is incompatible with life, has never been reported, in any case, So we need a little bit of protein. And we know that the more copies of SMN2 gene we have, the milder the phenotype it is. So you can see here, for example, that if there are 2 copies in green, it's more likely that the child will have a type 1. If there are 3 copies or more, it's more likely that the child will have a type 2 or type 3. So there is a sort of gradient. More copies of SMN2 gene are linked to a milder clinical severity. Okay, so this is a critical concept to understand also the effects of the medication. And in this picture, you see on the top, essentially, the 3 genetic mechanisms. So one is gene therapy that basically uses this viral vector. We are quite familiar with this for COVID. You remember all the pictures, this old 3D image. So this is like the external layer of the virus, the AV9, that uses the shuttle, okay, that is emptied of its original genomic sequence. It's emptied, bioengineered to include a single strand, a single sequence of the SMN1 gene. And this is basically injected into intravenously in the babies. to spread across all the body, particularly transfect the motor neurons, so that the machinery of the motor neurons, of the cells, can start producing the protein, the SMN protein. And then we have the two medication that actually use the other gene, the SMN2 gene, that essentially you see that the exon 7, is lacking in the SMN2 gene in the transcript, so in the passage from the DNA to the mRNA to the protein. And both nusinersen and risdiplam promote the inclusion of exon 7 into the transcript, into the mRNA, so that we can produce more SMN protein. You see here in the picture also the muscles, so we will go back to that in a little while, at the end of the presentation. So we have three wonderful old friends in the NHS that we can provide to our patients, of course with different criteria. As you know, gene therapy can only be delivered to type 1 babies or presymptomatic babies with 2 and 3 copies of the SMN2 gene, while nusinersen and risdiplam have broader label, of approval, but they are still, as it was mentioned this morning, under managed access agreement, meaning that NICE is still assessing the cost effectiveness of this drug for which patients, and which categories and so on. So this is why it's important to continue, as Maria Cristina was mentioning, the collection of this data. And shamefully, we don't have newborn screening, so we still see in the UK a lot of babies referred when it's too late. And this is why Giles was mentioned, the amazing work that also SMA UK is doing to increase awareness, to say, how many moms have told us, or grandmothers have told us, I had been worried for this baby since he was a few weeks old, and now it's 5 months, 6 months. And we have wasted a lot of time, because we know that it's critical to treat early to have the most of the benefits. So apart from these old friends, what's happening in research? Luckily, research is continuing because, as is what mentioned at the beginning, there is no cure, especially when we treat late. So I want to give you an overview of what's happening in research. This is a trial with nusinersen, high dose. It's called DEVOTE, because nusinersen was developed and capped to a certain level, to a certain dose, 12 milligrams. And you know that we need to do a loading dose, so for injection in the first 2 months, and then to continue the maintenance after every 4 months. And the question was, what about if we try to increase the dose, reduce potentially, the number of injection, and see if we can maintain, the same benefit, or even gaining some additional benefits. So this is what this trial is aiming to demonstrate. It was a double-blind, placebo-controlled, sham-controlled trial, meaning that some patients had the high dose, so 2 injections at the beginning, and these were in part B, were treatment-naive patients, infants, so babies with type 1 between 1 and 7 months, or symptomatic type 2 that were never able to sit between 2 and 10 years. And then there was a part C that was on patients that were not naive, so already had received other medication and basically switched to receive this drug. And again, the treatment was the 2 higher dose, so instead of the 4 loading dose, and then the maintenance dose of a higher dose instead of the 12 milligrams. It was 1 year, essentially nearly between 10 and 11 months of duration. And these are some of the data that have already been shared with the scientific community. So you see on the top, which is the gray here, the sham control data from the initial trial, because of course now it's no longer ethical to have patients who are not on any treatment. So they had to use, the historical sham arm of the previous trial that led to the approval of nusinersen. And you see that, what is neurofilament? Have you heard about neurofilament? So neurofilament is a biomarker, something that you can measure either in the CSF, when we take the lumbar puncture, or in the blood. And it's something that is not done routinely in clinic. It's mostly based on research to see if there is any sign of neuronal degeneration. It's an early indicator of the fact that the motor neurons are dying, are suffering. It's like when you do, a blood test to see if you have an heart attack or so. You're just a quick thing that you do from your blood. And you see, these are normally very high also in pre-symptomatic babies, especially with 2 copies, even before they display any clinical symptoms of the disease. So it's something that we can use very early to monitor if the disease is already there, without any clinical sign, that you can see in the baby. And it's something that is becoming part of the clinical trials with on pre-symptomatic babies, babies treated by after newborn screening. So the level were still high, so they tended to go down, but not significantly in the patients in the sham group, so patients that didn't receive at the time any medication, while there was a fast drop in the first 2 months, both in those who had the normal dose in the light blue, and in those, but more significantly in those who were treated with a high dose. So this was a sort of initial biomarker signal that you can have a faster drop in the neurofilament once you start the medication. So an initial signal, that basically you manage to stop somehow the progression of the death of the motor neurons. What about some clinical data, some efficacy data? As you see, the placebo group, so the ones that goes down, are those who receive the sham procedure in the initial trial, while in both the light blue and the dark blue, you see that both groups, so the normal regime with nusinersen, but also the high dose, continue in a similar way, so showed somehow similar improvement. And the same here is for the SMA2 children using the Hammersmith scale that we also do in clinic. So in terms of survival, you see that there is slightly better effect probably on the high dose that you see on the top, although the difference compared to the normal nusinersen was not significant. And you see here the Hammersmith in the type 2. So similar trend, let's say, of improvement, but with lower doses, with lower injection somehow, and some initial biomarker signals, of the faster drop that could potentially, give some additional benefit on the long term. But of course, we will need to continue, to monitor this. Another trial, that I want to share with you the results about is the rainbow fish. So this is with risdiplam that is, available through the NHS, but using in pre-symptomatic babies. And this is quite new because these are babies who have been identified by either newborn screening or prenatally and have been treated in the first few weeks of life. And these are the data of the 2-year follow-up. You see that there were patients who were with different copy numbers, and I think this is important to understand the results, the data. So you have patients who had 2 copies, so babies that are predicted to develop SMA1, as you know, with 2 copies, 3 copies or more copies that are predicted to develop either type 2 or type 3, so milder forms. This is important because when you go to compare different trials, different medication, you need to know this information. Okay, you cannot say this drug worked better than the other. You need to ask, what was the number of copies of this baby? At what age they started? What was the baseline levels clinical? Did they have symptoms when they started? Because this would affect the long-term effect, the long-term outcome, if the baby is already showing some symptoms. And for example, another biomarker that we use is the CMAP here. It's like the electrophysiology, that we use in research setting, but we are starting to do this in all pre-symptomatic babies that we see, because for us, even if the baby moves very well and is very active, and we cannot, with our eyes, see if there is any sign of the disease, neurofilament in the blood, CMAP can tell you the disease is already there. The disease is already showing something that is not clinically obvious, but is already there. And this could affect the outcome. And this is why we always say being identified with SMA through newborn screening or prenatal testing, especially when there are only 2 copies, is a medical emergency. There is no day to waste, okay? We need to start a treatment, any treatment, as quickly as possible. And these are some of the results at 2 years of this data, of this trial, pre-symptomatic babies. You see that the 2 copies here, all of them are achieved in the 2-year follow-up sitting. Some of them, most of them, also standing and walking. not all of them at the time of the last follow-up, 2 years, but of course the follow-up is continuing. While this was not a problem for the type, those who had more copies, 3 copies or more copies of SMN2, because all of them achieved all the milestones. So they achieved sitting, standing, walking, and as you will see here, in the dotted line, you see what is the typical time frame, for a normal child. You know that normally they sit by the age of 8 months or they walk by the age of 18 months. You see that the 2 copies here, many of them achieve sitting roughly, according to the timeline that is expected, but some of them require a little bit more, a little bit longer. Some of them require longer than the 18 months, to achieve standing or walking, and potentially they can still gain this motor milestone if we wait a bit longer. So the 2 copies again. And why is that? We need to look when they were treated. They were treated when they were 3 weeks, roughly 3-4 weeks. As we said, it's a clinical medical emergency. We need to start to treat early. So this may have affected also the outcome, why they have this delay in achieving this milestone. While this was not a problem for the 3 copies, all of them achieved the milestone sitting or walking, within the typical time frame. So not only they achieved this milestone, but they also did it according to what is expected. And this is why when we see that the child has 3 copies, even if it's identified by newborn screening or prenatal testing, we can probably say, probably we have a little bit more time, to act, because it's not so urgent to act as for the 2 copies. And the 4 copies or more, even more clear, they achieve everything, within what is expected, and they all achieved, the milestone. The good news is that also other aspects, not only motor, are assessed and are normal. So they speak normally, they develop good swallowing, they don't have any tube feeding or tube requirement, and they also don't require any respiratory support. So all the other domains also look very fine. And now we talk about what about, adding risdiplam to gene therapy. And there is a trial on purpose, because we don't have evidence at the moment. We don't have evidence, and we know that in the NHS we can only administer 1 medication. So there are 2 trials, Inhalia 1 and Inhalia 2, that essentially are looking at the benefits of adding risdiplam after gene therapy. And as you see, this needs to be before no more than 7 months, basically, after receiving gene therapy. Inhalia 2 is when you have a declining or plateauing phase after gene therapy, so you need to clearly see that there are signals of stagnation or deterioration, while Inhalia 1, the patients cannot be, can be doing well, irrespective, but again, they have this time window between the 3 months and the 7 months after receiving gene therapy. And the trial is recruiting, so we will see what will be the results in the future. Gene therapy, so we know that we can administer this to type 1 baby, essentially, or presymptomatic babies with 2 or 3 copies, but intravenously. Of course, there is a dose limitation. We know that the more we go up with the dose, the more is the risk of developing side effects, especially liver toxicity and other complications. So the approach of using intrathecal, similar to nusinersen, so the injection intrathecally, single injection of gene therapy or Zolgensma, can address the problem of limiting the side effects, of the intravenous delivery that goes everywhere, including the liver or other organs, when you can experience the side effects. And this is the trial in type 2, essentially, type 2 SMA, so this will be older than 2 years, and it was a trial in naive patients, so patients who were not treated with other medication, that were receiving basically the intrathecal and were type 2 patients, and these are the baseline characteristics. So they were roughly 6 years of age, as you see here, and they were either receiving the first year the intrathecal injection or not, and then they swapped to the other arm of the study in the second year. And you see that at the end of the 1 year observation, there was a difference. The blue is the patients who were receiving the intrathecal gene therapy, and the red is the ones who received the sham procedure. So there was a statistical significance, so a difference. And this is the kind of data the regulators want to see compared to the placebo, no? Because this is the best way to say a drug is working better than another one, no? Because there might be different confounding factors. And even other outcomes, not only motor function that we assess with the Hammersmith scale, but also the rule seems to be in favor of the effect of the drug. So positive signals, and indeed this drug is actually under consideration by FDA and potentially could become an option in the future also in the UK. Importantly, we were talking about the side effects, hepatotoxicity, the thrombocytopenia, the platelet reduction that we see in these patients immediately in the immediate aftermath of the infusion were very low. This is positive. The dorsal root ganglia toxicity, the reason that caused the pausing of this trial in the past, because we are going to inject this huge viral load in the back, in the spine, and the sensory roots that basically receive the signals, from the periphery could potentially be affected. And there were this data on the monkeys, that basically were showing that there might be some toxicity. So in this trial, there was no signal about that. And this was the trial where patients were already receiving other medication, nusinersen or risdiplam, and then they were switching to intrathecal gene therapy. Just again to show essentially primarily the safety profile of these are the baseline characteristics. So patients who were receiving either nusinersen for roughly 4 years, you see here, or 3 years of risdiplam, and then they switched to this medication. The safety was positive, meaning that nearly 30% have this drop in the platelets, while it's more common normally in the intravenous treatment. The hepatotoxicity was much lower. There were 2 cases of dorsal root ganglia toxicity, meaning like numbness or reduction of the sensibility, in the peripheral arms, that were not severe and were not didn't require any medication or any treatment and resolved with time, but something to bear in mind that is still there, as an option to consider when we use this medication. This is in terms of efficacy. So after switching, the fact that they show a sort of stability is also positive trend. They are not dropping in the score, so they were maintaining. They were even showing some improvement and then maintaining somehow because they stopped the other medication. So it's also a good indication. So this is another drug that potentially, could be considered for implementation in the NHS in the future for those patients who are not eligible for the intravenous gene therapy because of it's not approved. Now, combinatorial treatment, anti-myostatin, I'm pretty sure that many of you have already heard. So this is essentially, the myostatin is a protein, an enzyme that we have in our body to limit our muscle to grow too large, okay? Regulates muscle growth. And using something that stop or limit the activity of this enzyme, of this protein, is expected to promote further expansion of our muscle bulk. And the idea was, why don't we use this in neuromuscular diseases, like, for example, SMA? Because there were some positive aspects, data in the animal models. So this is the Sapphire trial from Scholar Rock. It's a phase 3 randomized, placebo-controlled trial where 2 doses, 20 milligrams and 10 milligrams, were used for 1 year in the patients. These are monthly infusions. And then there was a placebo group. All these patients, of course, were already on an SMN enhancing therapy. So they were continuing either their risdiplam or their nusinersen. And these were all type 2 or type 3 non-ambulant patients. This is essentially to show that patients were already on another treatment, and they were pretty similar, across the different groups. So you need to be careful that there is no difference. And these are the results. So the trial met the primary endpoint because they were looking especially at patients between 2 and 12 years. and at the difference in the Hammersmith scale between the treated group and the placebo group. And you see here that there was a statistical significance, and this is something that regulators, want to see to demonstrate if a drug is working, and also some other aspects like again Rulm or other positive trend in the older patients, older than 12 years, and you see here the difference with the placebo, with the patients who received either 10 or 20 milligrams in the number of patients who had at least 3 points increase in the Hammersmith. You know that we always look at these scores in clinic, and we say that 3 points is clinically relevant, when you show that there is a change. So you see that most patients in the treated group had at least 3 points of improvement at the end of the study compared to those who had the placebo. And again, also in other aspects like the Rulm, which is the scale for the upper limbs, So again, positive results. The trial is under consideration by the FDA and could potentially become another option to use in combination in the future. The good news is that overall it was safe and well tolerated, so there was no new signal, no new alarming signal in terms of safety. Roche is also developing this clinical trial using something similar, antimyostatin therapy in combination with risdiplam, also in ambulant patients, and the trial is ongoing. So the recruitment has been closed and they achieved the number of patients to be recruited, but the trial is still ongoing, so we will hear more in the near future. The last 2 slides. About this very important concept. You see the green dots here. These are the level of SMN protein, okay, that we need. On the left side is the prenatal time before birth. You see 2nd trimester, 3rd trimester. And then after birth, in the first 3 months, you start seeing a mild decline in the level of the protein. And then we need quite low levels, for the rest of our life. So this is the critical moment where we have to act. This is why we are advocating newborn screening. It is very astonishing that Scotland has beaten England, because they started newborn screening. I can't believe they can't speak English, but they started. They're starting newborn screening is unbelievable. So this is why, but potentially, I want just to open the door, the new frontier, especially for the 2 copies. Potentially we can have even more to use this therapeutic window. And this is the paper that has been published this year of a mom that has been treated prenatally of a baby with SMA diagnosed in utero, so treated with risdiplam towards the end of the pregnancy, and then the baby continued to take medication afterwards. And there is also this case at the bottom where they treated actually with something similar to nusinersen intramniotically in mice, so in sheep, so in mouse, in animal models, delivering similar to amniocentesis, with a needle, the drug, so that the baby can swallow and have the same effect. But this is still in the animal state. So a new frontier to use this therapeutic window, and I'm sure that Melissa may even further discuss about these aspects, and I'm happy to hand over to her.
Thank you, Giovanni. Hi, everyone. As Portia mentioned, I'm a lab-based researcher. I've been involved in SMA research for 21 years now. And I should define what preclinical studies is. At one point, I spoke for 10 minutes about preclinical with Portia, and she stopped me and she said, "What is preclinical?" It is basically anything before humans, okay? So it's using cells, and in SMA, we use a lot of animal models that we've engineered to have SMA, to study the condition, how does SMA impact different cells and tissues, but also to develop treatments. So the 3 therapies that are approved now for SMA were actually tested and developed in preclinical models before they reached patients. So we all know where we are now in the landscape of SMA, but there are a lot of research questions that remain, and we inform ourselves by what's happening in the clinic, by the clinical trials data, but also by real life experience that we hear from our clinical colleagues. But also we talk a lot with individuals with SMA and their families to understand what's important for them right now, and we bring those questions back to the lab. So I will give you a brief overview of kind of the hot topics right now in preclinical research. If we don't have time for questions later, because I know lunch is just around the corner, don't hesitate to grab me. I'll be here all weekend, and I've got a poster number 14 that you can grab me as well and chat about any ideas or questions that you may have. So one of the top questions right now is actually, can neurons regenerate? So we know that motor neurons are lost during SMA. We know that, the earlier the treatment, the better, but actually we don't know what happens to those remaining neurons. If we treat, do they maintain their health? Do they die back a little bit? Are they able to fully regenerate and rescue? And what's that time limit for that? At what age are we not able to rescue the remaining motor neurons? So that's one of the key research questions that's being addressed right now. This is just depicting what I've said. In SMA, we're losing that connection of the neuron with the skeletal muscle. And actually, early intervention and the SMN treatment, what happens to the neuron? Because we know that neurons have the ability to kind of grow a little bit and help out the muscles that have lost their connections with the neurons. But we don't know until what age that can happen, and if all the treatments actually enable the motor neurons to do so. So that's one of the research questions that's being addressed at the moment. One of the key topics that we're particularly interested as well, we've been working with Giovanni's team on that issue as well, are metabolic issues. What do we mean by metabolism? Well, what happens to the food that we eat? How do we store it? And how do we use it in energy? And all of the tissues and organs that we have in our body that allows us to store and use energy, like your liver, your pancreas, your muscle, how are they impacted in SMA? And how does that actually impact quality of life? And actually, once we understand those impairments, how can they be better managed? So we're thinking about various tissues. So we've got your pancreas that regulates your glucose metabolism, how you use and produce molecules to regulate your glucose, your liver, your brain is actually good metabolic tissues, your intestines, your muscle, fat. So, fat actually is a metabolic tissue, and that can be impacted in SMA as well. And heart. Heart's an important metabolic tissue, and I've seen a heart poster around. And when we look at kind of how we utilize, proteins that we eat, fats that we eat, sugars that we eat, how we utilize those are very different in the condition of SMA. So it's not just a matter of those tissues be impacted, but how we store that energy and how we use it seems to be quite different in SMA. And that's quite important, understanding how we can modulate that or how we can, help those impairments. And one of those ways that we're investigating, we've done some work with Giovanni's team as well, is diet, right? So diet is something simple that we can all change, when we're to impact our metabolism. So how does the SMA body work, and can we help it better by just tweaking a little bit the food intakes? Exercise. So I think there's quite a few sessions on exercise as well. So exercise impacts all of our metabolic functions. It's the same thing in SMA, but it might be different in SMA. So what we understand in a non-SMA individual might be completely different in the context of SMA. And drugs. And when I say drugs, usually they're drugs that are already available that are being used in other conditions, for example, in diabetes. Can they have an impact, for example, improving glucose metabolism? So those are all questions that we're addressing. I think an important one that's not talked enough is actually, can we improve the delivery of the current SMN disease modifying treatments? So they're not only being delivered in a certain fashion, but what's being used to deliver them can be changed and adapted to improve the delivery. So for example, there's the administration method, so whether it's an IV, orally, or through the central nervous system. but also the transport system. So that's an important one, for example, for the gene therapy, the AAV. Okay, it's a viral vector that transports the gene, but actually there's a lot of groups that are working on modifying that viral vector to make it a better delivery method for the SMN1. So that might impact what cells and tissues it gets in, but also if you need less of it because it's better, it also reduces toxicity. So a lot of groups are working on tweaking that AAV9, that viral vector, to deliver SMN, and also asking what cells and tissues do we want the drugs being delivered in. So depending on the drug and the administration method, you might be more targeting your neuronal cells, but might also want to make sure that the drug gets to your muscle, to your heart, and actually ideally to all the body, right? Because the SMN protein loss that leads to SMA is lost in all cells and tissues, even though the neurons are the most affected. And Giovanni's mentioned that as well. So can we deliver drugs in utero to maximize the benefits? So that's being investigated as well. So obviously you've shown two papers, one that was in the human population, but also in preclinical models of what happens if we deliver prior to birth, the gene therapies. And that requires some very skillful expertise of injecting embryos directly with the gene therapy, or also giving the drug to mom. So there's the two approaches that are being used right now. So you can either go directly in the embryo and also giving the drug to mom either through the water or orally, but both approaches are being investigated. Okay, can we use existing drugs to treat SMA symptoms? So I think we're all aware, we've talked a lot about cost effectiveness, so the 3 drugs that are on the market are very expensive. So if we're going to develop not new drugs to replace the existing drugs, but actually drugs that can help out to address some of the symptoms that are not necessarily fully met with the gene therapies. What we don't want to do is develop new drugs that are going to cost, again, in the millions, and that's not realistic. However, there's a lot of drugs that are currently used for other conditions that actually might be beneficial for SMA, and that's the most strategic way to develop new drugs because they've already been tested in humans for safety and efficacy, and so you're kind of bypassing that step, and a lot of them are commercially available. So this is kind of a busy, not slide, but a busy figure. But basically, to do that, what we tend to do is to look at cells and tissues of SMA mice and see, okay, what are the changes in those tissues at the DNA level, at the protein level? So all of those building blocks that lead to the creation of our body and our cells and our tissues, what are the changes in SMA? And then we use fancy computer software to allow us to match those changes to known activities of drugs. So drug X, we know it fixes all of these changes. So we identify a list of those drugs that are commercially available. However, we don't know if they're going to work in SMA, so we do have to test them in our labs. So we bring those drugs in our labs and we test them in our preclinical models, cells and mice, and see if we can improve some of the symptoms. Those drugs, those repurposed drugs, are never, ever as good as the SMN gene therapies. But when alone, they do improve some of the key phenotypes in our mice, such as weight loss and survival. It's an indication that we're in the right direction. And then the next step is usually, well, can we add this new drug with the SMN therapy and see if we have an additive effect, a combinatorial effect. If you run into that word, that's what we mean. And the idea is then there's shorter clinical trials, there's a shorter transition to individuals living with SMA. And then it was kind of like the new topic, I guess, in the last 5 years. So Giovanni's team is working on this kind of in individuals living with SMA. But the issue of cognitive issues in SMA has arisen. And actually, we're still in that phase of, are there cognitive issues? Are there not? What causes them? And, can we have more explanations of what causes them, and can we actually identify them quite early? So obviously we can study this in individuals with SMA, but we can also use preclinical models to study cognition. So we can use cells. So for examples, you can have human cells, human neuronal cells that you grow in a dish, and ask, okay, what does SMN loss do to these cells? Are there differences compared to non-SMA cells? But actually, you can use our mice. So the mice that have SMA, there's a different types of kind of learning and memory tests that you can do in mice and see actually if their learning and memory functions is similar in SMA mice compared to control non-SMA mice. And so those are the tests that are ongoing right now in the preclinical field. And once we understand what those impairments are in the mice, then we can backtrack and look at tissues and cells and try to understand at a molecular DNA protein level. Okay, and I've mentioned this. So can we improve symptoms by combining SMN-dependent and dependent drugs? So what we mean by that, the SMN-dependent are all the gene therapies, the nusinersen, Zolgensma, and risdiplam that are available. And independent are drugs that are not aimed at increasing SMN, but are aimed at improving other symptoms that occur in individual with SMA. So we've got our 3 depleted, and then we're trying to understand basically if we add either another drug, a diet, or an exercise intervention, can we improve symptoms overall, but also quality of life of individuals living with SMA. Okay, that was a roundabout tour. I'll leave it at that. If you have questions later, come and see me, but both of us look forward to your questions.
I'm sure there's lots of questions. Do pop your hand up if you have a question for Melissa or Giovanni.
Thank you both for insightful presentation. A quick question, all these clinical trials. What's your view of the time horizon? This is great. We're doing loads of research, clinical trials, but we know the battle then is to get it all approved and actually help the patients. So what's your gut feeling telling you about how long it might take to get any of this actually working and helping people?
Yeah, great question. I think that, we all know the strains that of the NHS, and the economical issues that all the countries have. In some countries we don't have any treatment or very limited access. I would say, following what we were discussing this morning, we need to continue to work together. All stakeholders, advocacy groups. My experience since I moved to the UK is that, as Maria Cristina was also mentioning, NICE is quite open, to hear the feedback. Even if there is an initial rejection, there is always, a second round of consultation. They want to hear, tell us about your experience. Does this drug work? Give us practical examples. So give us data, of course. They want to see the data. It's amazing if you manage to demonstrate, the effect compared to placebo. But even when this is lacking, what is the evidence that you have? What is the voice from the patients? Because sometimes we are not able to capture that. And then it's a matter to discuss and, agree on their levels in terms of the economical model. I think that, thinking about as a community, not only SMA in general, having a leverage to lower the prices of the drugs is also something good, that NICE has. And having this kind of back and forth, somehow may have the ultimate goal to negotiate, a better price for the system. But again, I see honestly a good level of openness to hearing, not saying no, we want to see the numbers, otherwise we will never approve it. But again, it's an ongoing process and we need to work together about that. But in the pipeline, there is the UK as well for many of these drugs as well. So we will hear more.
Thank you for your talk. My question is about biomarkers and whether there are any reliable ones you use in clinic. And I noticed you showed the slide about neurofilament, and it showed a 30% reduction with sham, which seems quite high. wondered if you could comment. Thank you.
I think it's something that we need to implement in clinic, and it's something that, especially for the babies who are more at risk of developing the more severe forms, could be an early indicator. It's something that we need to implement in clinic, but it's not routine practices still. In many labs, it's more common for ALS, for example, or other conditions. Because often, you lack the controls, the validation in the lab, but it's something that we are trying to use, because it will be very relevant when we will have newborn screening, because you can tell if a baby after birth, after being only a few days old, has already some, biomarker signals, and how quick you need to be, to act. This is helpful, I think, based on the evidence that we have so far in the conversation with the families, and how confident we might be in predicting the outcome. In my experience, still limited, because again, we don't have newborn screening, so the limited experience that I have in clinic is from moms who had another child and had a baby that was treated immediately after birth. This could be helpful, the CMAP as well. So having these biomarkers can tell you, okay, there is 2 copies, so the child has 2 copies, but these are still within limits, or not so elevated, or not so abnormal. So we may anticipate, based on the knowledge that we have so far, that the response to the treatment, if we start now, will be good and much better, compare if, for example, we have already some abnormalities or some mild clinical symptoms like reduced reflexes, a little bit of hypotonia, and so on. So it's something that can help, in the clinical practice in the future.
Can I come in on the biomarkers? Thank you, because I did realize this morning that I didn't have a biomarker slide, but that's an important preclinical study research aspect as well, because my understanding is that neurofilament is not optimal for SMA in terms of, when we say biomarker is something that you can measure that tells you information about disease severity, but also response to treatment that's easy to measure. So there's a lot of preclinical studies on that. And we have one that we undertook that's positive. There's no wood, knock on wood, but basically molecules that are released by skeletal muscle that so far, at least in our animal models, shows some correlation with disease severity. And also when we treat with gene therapy, it reduces as well. And then the next step for that is then to collaborate with clinical teams to confirm that in patient samples. Yeah. So thank you for your question.
Thank you for the presentations. Has risdiplam now become the sort of default treatment beyond nusinersen for children that either can't have the gene therapy or might be regressing? It just seems maybe that's the general direction of travel for clinicians at the moment.
We know that all the 3 drugs work. So when we discuss with families, we always say, the important thing is that we start the treatment now. Then there might be different criteria. It's more easy probably for the new babies to go for gene therapy, but we advocate for bridging, so start immediately risdiplam. But there might be patients who may not be eligible, for example, for gene therapy, and they may start with risdiplam and continue with risdiplam. There might be patients who are doing very well on one treatment, for example, risdiplam, and continue on that, for some time. So definitely, we don't have a recipe saying it's this or the other. Any of the 3 works very well if they are provided in a timely way. But then, there might be safety or other consideration, antibodies, for example. Some patients may have antibodies against AV9, so might not be eligible. So I would say that by default, we say start immediately, and then, you go for any treatment.
Are there any other questions? I'm not looking at them on the... I can give you a question that came up earlier, actually, that wasn't quite relevant for that session to finish us off. Someone was asking if there's any evidence that Zolgensma will last long term, or if you have any worries that the impact of Zolgensma might one day wear off.
Well, you can go and I can see the mouse data.
Exactly. So Yeah, I can start from the clinical perspective. Let's say that so far there is no signal that there will be like a weaning effect or a dilution effect over time, especially because motor neurons, once they are transfected with the virus, they are there, they are not like muscles that replicate. So, and we have now, the initial trial was like 9-10 years ago or even more, and these babies are still doing well. And we have even children who were treated presymptomatically with gene therapy, predicted to develop SMA, they are doing well. So after years of treatment, so without any other addition. So at the moment, there is no signal. And do you remember the last slide that I showed? So we need very high levels at the beginning and then very low level as a maintenance. So there is no signal that there will be a dilution or a weaning of the effect over time in the humans. Maybe you want to.
I'm just going to repeat what Giovanni said. In the mice, the one dose treatment at birth lasts, until the mice has reached 2 years of age, and there's increased activity in all tissues and cells that are looked at. Yeah.
I'm going to finish on a really controversial one. Which treatment's the best?
Me, I hate these. I am not a clinician. Well, it depends. I'm not going to answer that question, but I'm going to answer from a scientist point of view. We don't know what long-term viral presence in liver will do. That's all I'm going to say. But all three seem to have beyond positive results that we never thought we would see. Yeah. Oh, is that okay?
Yeah, perfect. Great.
Are you leaving it there? Thank you so much. Thank you. That was really interesting. And it's lunchtime.